Kia ora, my name is Patrick and my project is all about understanding weeds and pathogens in the high country, and my supervisor on this project was Jennifer Bufford. What I was aiming for was to answer my key research question, which is there a difference in the severity of pathogens affecting ruminant species across an elevation gradient? And by measuring across an elevation gradient, I could investigate how an increase in precipitation and a decrease in the minimum temperature as you go across elevation affects these plant pathogens. This meant I had to find sites at low and high elevations, such as this site here at Lake Sierra near Arthur's Pass. And on the individual scale, pathogens I was looking at harm the plant, as you can see here, by developing these spots across the leaf, which limits the photosynthetic capability of the plant. However, on the wider landscape scale, it is still poorly understood how pathogens affect plants. So what I was hoping to answer through my research question was sort of start to understand how diseases change across a wider landscape scale. The plants I was looking at for this project was Rumex obtusifolius here and Rumex crispus here. These plants are herbaceous, perennial, and are a common weed in disturbed areas, and they're also very widely distributed at both high and low elevations. The benefit of using these plants is that the pathogens affecting them are fungal with airborne spores, which means that there is easy transmission between populations. So each site I was likely to find at least some pathogens. Another benefit is these pathogens are genus specific. So by counting only the Rumex individuals at a particular site, I could find, by counting all of them, meaning that the pathogens only spread within the Rumex plants. And in order to get a wide range of elevations, I sampled both species at every 100 meters of elevation gain up to 800 meters. I sampled through Arthur's Pass on this transect here, Lewis Pass, up towards Lake Coleridge, and throughout Christchurch and the Port Hills. At each site, I sampled 10 plants per transect, and on each plant, I estimated the percent pathogen damage on 6 of the leaves using a transparent grid. I found that their elevation had no effect on pathogen damage, however, as you can see through the percent pathogen damage staying roughly the same across all of the elevation. And this was not what I had hypothesized, as I had hypothesized that as elevation would increase, then the percent pathogen damage would go up, as I would have expected that increased elevation would mean less suitable conditions for the Rumex plants. I also hypothesized that there would be other factors involved in determining pathogen damage besides elevation. So at each site, I also took other measurements. At each site, in order to calculate population size, I counted every Rumex individual within a 10 by 20 meter area, and along the transect for each plant, as well as estimating the pathogen damage, I also counted the total amount of leaves on each plant to determine roughly the size of each plant, as well as I noted the phenology of each plant, as in its life stages. For each one, I noted whether it was a rosette plant, where there would simply be a cluster of leaves growing near the soil, whether it was bolting, where there was a stalk growing out from the rosette as it starts to transition to the flowering stage, where it's beginning to show flowers, and fruiting stage, when it's beginning to show fruit. I hypothesized that an increase in population or individual size would result in higher pathogen damage, as there would be more available plant matter and the pathogen would be more easily able to spread between Rumex plants in the population. This was not what I found, however, as I found that these factors had no effect on pathogen damage. A potential reason for this could be that pathogens were already present from previous populations. This would mean that it would not matter that the size of the plant or the population may increase transmission within the population, as the plants would already be infected from these previous populations. I also hypothesized that as the older the plant became, the more pathogen damage it would experience. And while we did find that phenology had some effect on pathogen damage, it was not what we were expecting. I would have expected that rosette, the youngest life stage, would experience the least pathogen damage, and fruiting, the oldest life stage, would experience the most. However, what I found was that for both species, the flowering life stage had less pathogen damage, and for only Rumex crispus, the bolting life stage had less pathogen damage. However, our data does seem to show that there may be some pattern, in the sense that my data showed that there tends to be more pathogen damage towards the bottom of the leaves of the Rumex plants as compared to the top. And as Rumex plants transition from a rosette to a flowering, they tend to drop a lot of their bottom leaves, which points to this pattern here. And as they transition from flowering to fruiting, this would have allowed enough time for the pathogen damage to build up again. As for only Rumex crispus having bolting individuals and not Rumex obtusifolius, a possible reason for this could be that Rumex crispus plants tend to develop quicker and drop their bottom leaves more quickly than Rumex obtusifolius. So in conclusion, I found that individual size, population size, and elevation had no effect on pathogen damage, but phenology had some effect on pathogen damage. Thank you for listening, and thank you for my supervisor, Jennifer.
Questions for Patrick?
Yeah, the pathogen damage you're showing seems quite low. And I'm just wondering, I guess two things. One is, do you think it's actually a problem for plants and limiting them? And do you think maybe the lowness is related to being fairly early in the summer?
That could be. What I found sort of across the board, and even for that one, that's probably quite high compared to what I found. So probably quite similar. It probably doesn't affect it greatly, and potentially it would be quite interesting to sort of do it at different times of the year and see if the pathogen damage would be worse at other times of the year.
Sarah?
Just wondered if you, I know you didn't identify the pathogens specifically, but do you have a feel for whether you were looking at different things at different points along your elevation gradient?
It appeared that across the entire elevation gradient, the pathogen damage stayed sort of visually very similar, but in order to properly isolate which ones, I'd have to go through genetic sequencing. And also from previous research, I learned that initially I tried to isolate them and sort of look at them through the microscope, but it was sort of almost impossible to tell, and I would have needed a lot more time doing genetic sequencing and properly isolating them. So.
Thank you, Patrick. Okay.